Good morning, everyone. Today I'll be giving a lecture on microarrays for BSc Biotech third year. Um, the subject being your RDNA technology. Now, what are microarrays? Microarrays are again a type of series of probes which are present, which we use on a solid substrate to hybridize different types of target DNA. Say I have to find out a DNA target DNA. What I'll do is I'll make a use of a microarray in that case. What is a microarray? They've been used many times. They've been used in different types of things which have happened. The first case when they were used when in case of yeast, which had around 6,000 genes, we used these microarrays there. Now the microarrays consists of two parts. One is probe, the other one is target. The probe is, what is a probe? Probe is a small nucleotide sequence which is having a known sequence basis on them. It is fixed either onto a solid substrate to form an array, or what you can do in that case is whereas the source, in case of source, you've got a purified mRNA, isolated DNA, or a complementary cDNA, complementary DNA, along with some ESTs, whereas short sequences are obtained from the ends of cDNA. So that is something which is there. Then there is target. Now in case of a target, this is a DNA sequence which has to be studied. Say it can be a DNA sequence which we have either isolated, or we're trying to find out the gene for that thing. So that is something which is there to be studied for a position, composition of the gene, or for the gene expression, or for a mutation which has happened in a gene. That is what we can try to find out with the target gene which is there. Now then we have got we either label these are probes. The labeling can be done either with the fluorochromes, or you can have other labeling components which can be there. They can be either radioactive or something else which can be used. Now, probe and the target in this case is the opposite to that in the southern blot experiment. Now, in this case, you've got a different thing. Now, target is fixed. In southern blot, it was the other way around. Here, the target is fixed onto a nylon membrane or a nitrocellulose membrane, whereas a probe or a labeled DNA solution is generally added from outside. So that is something which is there. Now, types. Now, there are two types of microarrays which are there. Now coming to the types which are there, the first one, the type that is the DNA microarray, the other one is the antibody microarray. Now first I'll be talking about the antibody microarray. It consists of, what in this case we do is, there are spots of different antibodies which are there, and these are used to measure the abundance of different proteins in a sample. So you can go in for checking the proteins in a sample. Now DNA microarray, in this case what happens, the DNA sequences representing genes of an organism, say a microorganism, are spotted into a slide. These slides are used to determine the genes using a transcribed in the cells or the concerned cell. That is the transcriptome. Basically, the transcriptome is what is we are studying here. What is the transcriptome? Transcriptome is something which is called as the sum total of total mRNAs which are present in a cell. Now coming to the DNA microarrays, they can be there can be further of two types. One is the cDNA microarrays, whereas the other one are the oligonucleotide arrays. Now cDNA microarrays, so what happens in this case? Probes which are cDNA here are blotted onto a gene. Yeah, they are plotted onto a gene and substrate with the help of a fine needles. You use fine needles and you plotted them onto a substrate or with a robotic arm. Generally, this thing is done with a robotic arm. You have a chip, onto that you fix up these things. Then you have got the oligonucleotide arrays. Again, what are these? These are probes which are oligonucleotides formulated in situ. You have to produce them there at that point itself, directly onto the chip. You put these oligonucleotides directly onto the chips, and then they are immobilized. You can immobilize with any method. Either you can use a sodium arginate or something else onto a thin wafers or a silicon glass, but different techniques like photolithography. You can use a photolithographic method for fixing up these things there. Now coming to the features, I'll be telling you what are the features which are there in case of cDNA and oligonucleotide arrays. First one is the feature here is the source of DNA. In case of cDNA, it is the cDNA itself, cDNA microarray, it is the cDNA which is the source of DNA probe. Whereas in case of oligonucleotides, what we do is the oligonucleotides are synthesized directly onto the silicon wafer, the wafer which we are using for the probe. The length of the DNA probe, either a whole gene or a part of a gene can be there. Whereas in case of oligonucleotide arrays, what you happens is 20 to 30 nucleotide, they're present. Now abundancy of probes, about 30% of, in case of cDNA, about 30% of cDNA probes are redundant, they are useless. 30%. Say I've got 100% of that, about of 30% of those they are redundant. Whereas in case of oligonucleotides, we are producing them in situ, so there is no chance of any redundancy which is there. So as we know, each oligonucleotide sequence is chosen to represent a different gene. So there is no chance of redundancy here. Now, cross hybridization, that is a problem in case of cDNA. You can always have a cross hybridization taking place. Whereas in case of oligonucleotide, the cross hybridization chances are very less, and they're minuscule in that sense. So what happens in this case is 93% you got a success rate which is there in case of oligonucleotides. Now coming to the principles of the microarray, what is the principle behind working of these microarrays? The principle of these DNA microarrays technology is based on the fact that the complementary sequences of a DNA can be used to hybridize an immobilized molecule. So I've got an immobilized molecule, a complementary cDNA will go and attach with the molecule itself. The process of the hybridization, the double-stranded DNA is there. They're subjected to conditions like alkali solution or a sodium hydroxide solution. You can use any alkali solution so that the hydrogen bonds between the two bands which form the phosphodiester bonds between the DNA strands remain intact, leading to the production of two separate bands. The phosphodiester bonds are always there, whereas the hydrogen bonds they are broken up, so that you have got the two strands which are there. When treated with an appropriate probe, it results in, if you put up a probe into this, say I've got a complementary DNA, I put up a particular probe onto that. What happens in this case is you've got a pairing of the probe with a complementary base, and the target DNA can be found out. So this is the target DNA, and this is the sequence which I'm trying to find out. I've got a probe. This probe will come and fix only here, not there. It will fix at a particular point itself. So what you can have is that probe can go and find out the particular sequence which is there on a complementary DNA or on the DNA as such, what is there. Now, DNA sequence of each gene to be analyzed of an organism. Say I have to analyze an organism's every gene. So in this case, they are labeled with fluorochrome. You can put up a fluorochrome, any fluorescent dye you can use, and they are labeled. And what you can do is you can go in for hybridization is initiated. Generally, there are different steps which are involved in the hybridization. Double helix open up and hybridize with a complementary DNA, and in producing a fluorescent at the hybridized site. जहाँ पर जाकर लगेगा, you'll have a hybridization happening there. And say there is a fluorochrome which is there, that dye, when you put up a radiation onto that, either a light or something else, or even a UV radiation, what you'll see is there is a spark which is going to happen there, and you can see your particular site. So these can be scanned, these can be quantified, and you can correlate with these things. Now, what are the steps of microarray experiments? There are different steps. I'll be dealing with these steps tomorrow. Means yesterday. We have already talked about these things. Tomorrow I'll be continuing with the same lecture here. Thank you.